So we know in our genome we have genes. And we know these genes can be transcribed into mRNA. Then these mRNA can leave the nucleus and be translated into proteins. And that's what genes do. Genes are used to make very specific proteins. But how does this process work specifically? How does this transcription process work? Well, let's say we have a chromosome. In this chromosome, we know we have our gene. We have our gene, which is just a nucleic acid sequence, a very specific nucleic acid sequence, which can be used to make proteins. But usually we don't just have a gene. We'll normally also have a promoter. We'll usually also have a promoter, and not just that, we'll also have an enhancer. And sometimes this enhancer can be thousands of base pairs upstream. So I use this symbol to show that there could be thousands of base pairs in between. So what are these components and what do they do? And also, just a little convention, the first nucleic acid that's transcribed into mRNA, we call it the +1. Then the second one that's transcribed is +2, the third one is +3, etc. So that's just convention. So then this +1, the first one that's transcribed, the one adjacent to it would be -1, and then this would be -2, -3. And then so this would be downstream, going towards higher positive numbers, and this would be upstream. And then normally they're around 25 nucleic acids upstream is when we have the promoter. Then once we have the promoter, usually it's thousands of nucleic acids upstream until we get our enhancer. But now we have this gene. So what determines whether this gene is activated and being expressed to make a protein? Because just because a cell has a gene in its genome, that doesn't mean that gene is actually used. Sometimes the gene can be inactivated and not used. So what determines whether a gene is actually activated and being used to make proteins? Well, all genes have transcription factors, and these transcription factors regulate whether this gene is being expressed or not. So if this gene has a transcription factor that's inactivated, then this gene is inactivated and it's not being expressed. However, what if we get some kind of signal that activates this transcription factor? Now the transcription factor is activated, now can activate the transcription of this gene and activate this gene. So the transcription factor will bind to the promoter. So it'll bind to the promoter. And normally these promoters have these TATA boxes, which are essentially just TA nucleic acids, but the transcription factor is activated, so now it binds to the promoter. Once it binds to the promoter, now it can recruit an RNA polymerase. And once we've recruited an RNA polymerase, now can start polymerizing our mRNA using our gene as a template. So again, the point is we activate the transcription factor, now it binds to the promoter, which can recruit an RNA polymerase, which can now we're activated and we have everything we need to transcribe this gene to make mRNA. But sometimes we can go a step further. For example, I'm just using cortisol as an example, but let's say some cortisol comes along. This cortisol can bind to the enhancer. Specifically, in this enhancer, it will bind to a cortisol response element, which is just a nucleic acid sequence in the enhancer. So the cortisol will bind specifically to the cortisol response element. Now what happens? Well, now essentially this chromosome will bend. And remember, there's thousands of nucleic acid sequences here. So essentially, we would bend. And now what would happen is the cortisol will start recruiting other proteins. And once these other proteins are recruited, now we have this complex, this protein complex, which highly upregulates transcription. And now we're going to do a lot of transcription. So the point is, these enhancers enhance transcription. And again, I just use cortisol as an example. Maybe estrogen comes along. And binds to an estrogen response element, which forms this complex which upregulates the transcription of this gene. But in reality, all we need is the transcription factor, and that's all. As long as we have an activated transcription factor, that's sufficient to recruit RNA polymerase and express this gene. Enhancers can enhance the expression of this gene, but they're not required. So we see we have these transcription control regions, which regulate how much transcription we have. But if we have an activated transcription factor bind to the promoter and recruit an RNA polymerase, now we have everything we need to transcribe this gene. So now we can transcribe this gene, making our mRNA. But something important to realize is this chromosome is double-stranded. So because it's double-stranded, it always has a negative sense strand and a positive sense strand. And sometimes the negative sense strand is also called the antisense, and sometimes the positive sense strand is also called the sense strand. So we have this polarity. We have these two strands which technically have different nucleic acid sequences. So which strand are we going to use as a template? Are we going to use the negative sense strand as a template to add complementary base pairs to make our mRNA, or are we going to use the positive sense strand as the template to add complementary base pairs? Which strand do we use? Well, I'm going to tell you now. We use the negative sense strand, and that's why the negative sense strand is also sometimes called the template strand. So this gene is double-stranded. We have a negative sense strand and the positive sense strand, but it's the negative sense strand that we use as a template to add to make our mRNA. So now we make our immature mRNA, and sometimes it's also called hnRNA. But for this video, I'm just going to call it the immature mRNA. So we use our template strand as a template to add these complementary base pairs to make our immature mRNA. And will this immature mRNA be considered positive sense or negative sense? Well, it would be considered positive sense. It's positive sense because it's complementary to the negative sense strand. And I know this is a little complicating, but the point is we have these two polarities. It can either be negative sense with this negative sense sequence, nucleic acid sequence, or this positive sense nucleic acid sequence. And again, we use the negative sense strand as a complement to make our immature mRNA, so the immature mRNA is the positive sense. And we know it's positive sense because it's the exact same sequence as the positive sense strand. The only difference is because this is DNA and this is RNA. Instead of T's, we use U's. But again, we make our immature mRNA, which is positive sense. So now we have our immature mRNA, our transcript, our original transcript. So this original transcript has certain regions. For example, in the 5' region, it has a 5' untranslated region. And in the 3' end, we have a 3' untranslated region. And remember, we always make mRNA from the 5' to 3'. So again, we were adding it in this direction from 5' to 3'. So again, we have a 5' tail and a 3' or a 5' end and a 3' end. And again, on both ends we have these untranslated regions, and they're called this because these regions do not get translated. These codons don't get translated to make amino acids and proteins. So that's why they're called untranslated regions. So internal of that, we also have these exons and introns. And later on, I'll explain what these exons and introns are important for. So we make our transcript with these different regions. But now we do some processing where we make this transcript a little more mature. And the way we do this is we add a 5' cap and we add a 3' tail. And normally what would happen is in the 3' untranslated region, there'll be some kind of sequence that acts as a signal that tells the cell to add a 3' tail. And I didn't draw the sequence, but the point is there's a sequence in this 3 UTR that tells the cell to add a poly A tail on the 3' end. So now that we have this transcript, now essentially what we can do is we can go through a process referred to as alternate splicing. So essentially these exons will always keep. We never get rid of these exons. However, we can selectively get rid of these introns. For example, maybe we get rid of this particular intron. So if we specifically get rid of this intron, we know these regions will bind, and when we get rid of this intron, we essentially would make this guy. So this is referred to as mRNA. This is a mature messenger mRNA. This is what we're referred to as mRNA. Once we have the 5' cap, the 3' tail, and we've gone through splicing, now this is called an mRNA, which can be translated to make a very specific peptide. So again, we could get rid of this intron, but also maybe instead we get rid of this intron. So what happens if we get rid of this intron and these two regions bind? If we do that, we'd make a completely different mRNA, where again we get rid of this one. So if we get rid of this one, we'd essentially form this mRNA, which can be translated to make a completely different peptide. So notice the utility. We can take one gene. We take this one gene, making this transcript, and we can use this transcript to go through alternate splicing. For example, if we get rid of this intron, we'll make a very specific mRNA, which will make a very specific protein. However, if we get rid of this intron only, we'll make a different mRNA, which will make a different protein. Or maybe we get rid of both introns. If we get rid of both introns, we make a completely different mRNA, which will make a completely different protein. So notice we can have one gene be used to make several different proteins. And that's the utility of this alternate splicing. But again, let's say, for example, let's say we get rid of this particular intron, we'll make this particular mRNA, which will make a very specific peptide. So now we have our mature mRNA, which can essentially enter into the cytoplasm. But you might wonder, why? And again, we know it's the mature mRNA because we have the 5' cap, we've gone through splicing, and we have the poly A tail. So that only once we've gone through all those modifications do we have an mRNA. But you might wonder, why do we have this poly A tail? Well, once this mRNA enters the cytoplasm, there are these exonuclease enzymes which essentially are degrading this mRNA from the 3' side. Starting from the 3' side, it starts degrading this mRNA, cleaving off one base pair at a time. However, if we have a very long poly A tail, then it will take a long time for this enzyme to degrade and chop off these base pairs. So if we have a very long poly A tail, it'll take a long time until we chopped off enough base pairs to start getting into the coding region. So essentially, the longer the poly A tail, the longer the stability of this mRNA, the longer this mRNA will be around to be translated into proteins. So if we have a very long poly A tail, this mRNA will last for a long time until this exonuclease starts degrading the coding region. So again, that's the utility of the poly A tail. It essentially makes this mRNA have a longer half-life, and it makes it more stable. But what about the 5' cap? Well, we know this mRNA will bind to a ribosome so it can be translated to make proteins, and it's specifically this 5' cap that binds to the ribosome. So that's why this 5' cap is important. It binds to the ribosome to allow this mRNA to get translated. And again, the 5' cap and the 3' tail also have other purposes, but these are some of the major purposes. So this is how eukaryotic cells transcribe genes. That's how we take this gene and these transcription control regions to make our transcripts, to make our mRNA that can then go through translation.